Hello everyone, and thank you for joining us. Welcome to today's webinar titled Modeling Human Gastrointestinal Development and Disease Using Pluripotent Stem Cells. My name is Jenny Marie Peterslund. I'm a Product Marketing Manager at STEMCELL Technologies, and I'll be your moderator for this webinar. And it is now my great pleasure to introduce you to today's speaker, Dr. James Wells. Dr. Wells received his PhD in genetics at the State University of New York at Stony Brook before going on to his postdoc at Howard University with Dr. Douglas Milton. He became an assistant professor at the Cincinnati Children's Hospital Medical Center in the Division of Developmental Biology in 2002 and has since dedicated his career to understanding the processes by which gastrointestinal and endocrine organs form in the developing embryo. Guided by these developmental processes, Dr. Wells and his lab have pioneered the differentiation of pluripotent stem cells to gastric, intestinal, liver, and pancreatic cell types, and recently to intestinal tissue in organoid culture. Thank you, Dr. Wells, for taking the time to present to us today, and let me now hand over the presentation so you can begin.
Thanks, Jana Marie, and thanks to Nick for organizing this. Okay, well, thank you all for attending or listening, and I'm happy to tell you about some of our work developing human gastrointestinal organoid systems. First, I want to start by thanking the lab. I work with a really fun group of trainees and fellow faculty members here at Cincinnati Children's. And I want to start just by giving you a brief rundown on different gastrointestinal organoid models that are out there. One that you're probably most familiar with was really pioneered by Hans Clevers and colleagues. And in these types of gastrointestinal organoids, patient biopsies are obtained and primary epithelial cells are grown in culture into either gastric or intestinal or colonic organoids that can be used for a variety of applied studies. Now, the types of organoids that my lab has developed are quite different in that we start from cultures of pluripotent stem cells and differentiate them into organoids using directed differentiation strategies that are based on normal organ development during embryogenesis. And both types of organoids are useful for many of the same things, but also have some unique uses that are peculiar to their own, the way that they were derived. So for example, I'll focus my talk on organoids that are derived from pluripotent stem cells and how we're using them in a variety of ways to study human development of organs and congenital diseases that affect organs, how we're using them to engineer more complex tissues that we can test in various transplant models, how we're using them just to currently and in the future for physiology and drug response, and how we might use them to study how various pathogens that affect the GI tract can be studied in vitro or in vivo. Now, and today I'm going to focus predominantly on human intestinal organoids and human gastric organoids and how we derive them from pluripotent stem cells and how we're just in the early stages of trying to use them to study various human diseases of either the stomach or the intestine. Now, one reason we focused on human pluripotent stem cells are that pluripotent stem cells have unlimited capacity for replication and they can become any cell type of the body. What that means, of course, is that any disease that affects any cell type or tissue type can be, those tissues and cells can be generated from pluripotent stem cells. And we're using human because, really for the first time we're able to study human developmental biology using pluripotent stem cells as a model. I'll try to convince you that using pluripotent stem cells we can generate complex tissue structures and then, as I mentioned, use them to model disease. Now, what's really special about pluripotent stem cells is that they can be generated from any patient that walks in the door using the Yamanaka-based induced pluripotency paradigm. So therefore, any patient that comes in with a specific genetic disease can be modeled using pluripotent stem cell-based approaches. And as I mentioned, these are all human, and that's important because in many cases, cells in humans don't behave exactly the same as they do in other animal models. So in some cases, we might get better disease modeling if we generate human tissues. However, the biggest challenge for using or for generating pluripotent stem cell derived tissues is understanding how they're made during embryonic development. So a lot of the talk will go back and forth between developmental biology of organogenesis and generating human organoids using those developmental paradigms. And the first example I'm going to talk about is really to introduce you to the concept. This was the first organoid that we developed. This is developing small intestinal organoids, which was published a number of years ago and was really a great collaboration with Jason Spence when he was a postdoctoral fellow in my lab. He's now running his own lab at University of Michigan. And then more recently by grad student Kyle McCracken. So, of course, to understand organ development, I'm going to have to give you a bit of a primer on organogenesis in embryos. And I'm not going to go into a lot of detail, but suffices to say that organs are developed in a stepwise process. And on the left panel here, you see different stages of mouse embryos that, starting from right after gastrulation up through about the stage where you can start to actually visibly see organs emerging. And we're focusing on this step because trying to generate any of these organs down below through a directed differentiation of pluripotent stem cells will have to rely first on making endoderm. Endoderm gives rise to the entire epithelial lining of your gastrointestinal tract. We're then going to have to understand how to push endoderm into specific organ types through a basic process called patterning. Endoderm, which starts as a two-dimensional sheet of cells, has to form a three-dimensional tube. And in the embryo, this three-dimensional tube extends from the developing mouth of the embryo all the way down to the posterior of the embryo. And that endoderm tube will give rise to all the different organs that you see below here. So within 2 days in the mouse, after gastrulation, you can already see the emergence of very definitive organs forming. So for example, you can start to see the liver forming, the stomach forming, and this tube down here will form the small and large intestine. So this 2-day window of time is really quite pivotal in how you establish initial organ development. And has really led the way for us to understand how to push endoderm derived from human pluripotent stem cells into these different organ derivatives. So I also want to point out that this arrow popped up here because now we're using cultures of human pluripotent stem cells to do primary research and discovery on how these different organs form in vitro, in that these stem cell in vitro models are very powerful to do high throughput screens for manipulation of different signaling pathways, and that after we discover, for example, basic fundamental strategies of how organs form in these in vitro models, we can actually go back into model systems like mouse to test if in fact these organs form according to our in vitro paradigms. So effectively these in vitro organoid systems are now their own research tool to study basic concept of organogenesis. Now, the first example of generating intestinal organoids was really done based on previous studies both from our lab, but also many other labs who have pioneered the basic understanding of how these different organs form in the embryo. And through many great collaborations, including colleagues who were here at Children's Hospital, Aaron Zorn, and formerly Noah Schroer, who used to be here at Children's, we've taken all this published information and applied it to directed differentiation of pluripotent stem cells into intestine. And the first key step, which was actually discovered by a number of other labs, including Gordon Keller's lab and ViaCyte, discovered that you can make endoderm and you can direct differentiation of stem cells into endoderm by activating a particular pathway in early stage cultures. And this is done through manipulation of nodal signaling with the nodal mimetic activin. But at the end of the day, what that means is that pluripotent stem cells can robustly differentiate into definitive endoderm, and in our hands this is shown to be quite efficient, 90% or so positive cells are definitive endoderm. However, it should be noted that many of these cultures also still retain some mesoderm cell types. Now, endoderm, which can form any of the epithelium of any of the gastrointestinal organs, then we have to direct it into specific organ subtypes, and we do this through an embryonic process called patterning. And all that really means is that if we want endoderm to form intestine, we have to push it into forming posterior endoderm, which we did through the manipulation of the FGF and Wnt signaling pathways, which our previous studies had shown these pathways are essential for turning endoderm into more posterior endoderm. And we read that out by looking at expression of the key posterior determinant, the transcription factor CDX2. So this shows that FGF and Wnt can very nicely push endoderm into a posterior endoderm, which was expected from our embryonic studies. What was not expected is that the combined activity of these two pathways also coaxed two-dimensional cultures of endoderm to form these three-dimensional structures that we're calling gut tube spheroids. So at the end of the day, we got transition into three-dimensional gut tube that is posterior, which is, of course, the sort of precursor structure to the intestine. Merely by moving these to three-dimensional cultures in the presence of a EGF, which just helps their growth, we can actually grow these small spheroids out into human intestinal organoids. Now we know these are intestine, shown in the next slide, because they express key intestinal markers. This intestinal organoid here, which is a couple of millimeters in diameter, has an epithelia that is all CDX positive and has absorptive enterocytes, goblet cells, Paneth, light cells, and enteroendocrine cells, which are the main cell types of the developing fetal intestine. Just schematically shown here is a cross-section of the intestinal epithelium. So they not only have different absorptive and secretory cell types, they also have some rudimentary absorptive and secretory function. So here's just a three-dimensional image of an organoid. In this case, what we had done was inject this dipeptide into the lumen of the organoid. And what you can see here, the little white cells are actually enterocytes that have taken up this dipeptide, which means that they have an active dipeptide transport system. Now the other thing that the intestine likes to do is to secrete, and two things that it secretes are mucins into the lumen. So that this is an organoid that's secreting this green mucin into the lumen. But also these organoids can secrete hormones, which is another key job of the intestine. Only in that case they secrete hormones basally. Now while these small 2-millimeter organoids have some rudimentary function, their structure is really quite primitive. However, what we found is that if we take one of these organoids and give it a blood supply, and how we do that is we engraft it into an immunocompromised mouse, and the mouse vasculature will actually plug into the organoid, and after actually 6 to 10 weeks, we harvest and look at them. What you can see is that a 2-millimeter organoid will grow into centimeter-sized tissues. This is an organoid that was grown on the kidney, and this is an organoid that was actually engrafted into the intestinal mesentery. Now, we're in the process of developing different tissue transplant models in collaboration with Mike Helmrath, and here's an example of one of the models that Mike and his lab mates have developed. They're, of course, in the process of developing a number of other transplant models, for example, one in which we can anastomose human intestinal segments into the mouse intestine. But that'll be for a future webinar. But in this case, if you just look at how well these organoids mature after a number of weeks in vivo, by sectioning through that human tissue, you can see that all that tissue is well and truly human. It has human nuclear antigen, and you can see that the epithelium has nicely matured into structures that resemble villi and crypts, shown here. You can actually show that these crypts have stem-like cells, intestinal stem-like cells, by virtue of their expression of this intestinal stem cell marker, LGR5. I also want to point out that the organoids that we generate in vitro not only have epithelium, they contain a mesenchyme. And following growth in these transplant models, we see nice differentiation of the mesenchyme into layers of intestinal smooth muscle. Here in green is shown submucosal muscle, and this layer of muscle out here are the layers of my enteric smooth muscle. So at the end of the day, you have a very complex structure with epithelia that have villi and crypts, and mesenchyme that have differentiated into smooth muscle. So to summarize the intro, I think that through manipulation of basic developmental pathways in a temporal manner, we can generate intestinal organoids that have all the properties to go on to form nice epithelia and mesodermally derived structures. And to transition into the second part of my talk, I want to tell you now what those organoids were lacking. Now, they had nice secretory and absorptive function, smooth muscle, but what they were lacking was the ability to undergo the coordinated muscular contractions called peristalsis. Reason being is that they were lacking the nervous system or the neural component of the gut that innervates the myenteric and submucosal layers of muscle to drive not just peristalsis, but also the neurons of the GI tract control other key functions like barrier function and hormone secretion. So we next went on to try to devise a way that we could generate tissues with more complexity by engineering organoids with an enteric nervous system. And again, using developmental principles to do that. So the enteric nervous system, most importantly, this was done as a great collaboration. My student Mike Workman really helped to pioneer this approach, but we couldn't have done it without our great neural crest cell experts, Sam Brugman and Cheng Fang, and of course, ongoing collaborations with Mike Helmrath and Maxime in Mike's lab. This work was recently published just this year. So the enteric nervous system, as I mentioned, drives peristalsis and is important for barrier functions and whatnot, and it does so through the coordinated activity of over 500 million neurons and glial cells that innervate the entirety of the gastrointestinal tract. Because of the complexity here, it's been now termed the second brain for people. And defects in ENS, enteric nervous system, can be debilitating and result in severe GI dysmotility issues like Hirschsprung's disease. So where does the enteric nervous system come from? That's kind of an essential question we had to ask ourselves if we wanted to engineer organoids with an enteric nervous system. So where it comes from is, in fact, if this is a cross-section of an early stage embryo, just kind of a generic cross-section, where the neurons of the gut come from the entirely other end of the embryo, the dorsal end of the embryo, from a structure called the neural tube. Now, cells that give rise to the enteric nervous system delaminate from this neural tube and form a cell type called neural crest. Now, these neural crest cells migrate all the way down to the ventral side of the embryo, which is where the developing gut tube is just forming, and they actually migrate into the gut and differentiate into the neurons and glia. So how on earth are we going to get this complex embryonic process to happen in these organoid cultures? Well, what we did is pretty simple. In one dish, we generated neural crest cells using an established protocol from Bajpai et al. And we used these neural crest cells and incorporated them into our developing organoid cultures. Practically how we did that is seen in the next slide. Literally, we generated early stage intestinal organoids on the top panel here, and then neural crest cells separately, and combined them. And just through mechanical aggregation and centrifugation, the neural crest cells were recombined with the early stage organoids, put in three-dimensional matrigel cultures, and grown out into in vitro derived organoids. Now, we either studied them, the in vitro derived organoids, or we transplanted them, like I had just shown you, into the mouse to grow and mature them into something that is more reminiscent of enteric nerves. And I'm going to just give you a brief summary of our approach here, and I'll show you some of the data after that. So firstly, when we recombine the neural crest cells and the early stage organoids, very quickly the neural crest cells migrate into the developing intestinal mesenchyme in vitro, very similar to how they do it in vivo. They differentiate even in vitro into a variety of different neurons and glial cell types. And they have some rudimentary function, which I'll show you through some live imaging. Moreover, when we transplant them into mice, they form a three-dimensional neuroglial plexus that contains functional interstitial cells of Cajal and can have neuronal-mediated peristaltic-like motility. Data that I won't show you is that these integrated neurons and glia actually have a coordinated neuromuscular coupling with smooth muscle and can control contraction and relaxation. So let me show you some of this data. Firstly, to show that the neural crest cells migrate into the mesenchyme, we compare neural crest cells in our in vitro engineered organoids with the real deal. This is a cross-section of a gut tube from an 11.5-day mouse embryo. Now, the red donut in the middle here is the developing intestinal epithelium. All the little green speckles on the outside are neural crest cells that have differentiated into neurons. And you can very easily see that down below, our engineered organoid epithelium is surrounded by these differentiated neurons that derived from neural crest cells. So they have a similar architecture even at early in vitro stages of differentiation, similar to actual developing intestine. I'm next going to show you a video showing that they have some rudimentary function, and this was done using a GCamp reporter. And for those of you who don't know what GCamp is, it's actually a reporter that in live real time can measure efflux of calcium into a cell. And when calcium effluxes into a cell, this fluorescent reporter fluoresces. And it's actually done in real time. And this was a pluripotent stem cell line that was given to us from Bruce Conklin at UCSF. So the effluxing neurons, you know, that's not entirely satisfactory in terms of really forming a neuroglial plexus like you'd find in this cross-section of the gut. So what we did to see if we could get a neuroglial plexus is we took those in vitro derived organoids, and like I said before, we engrafted them into mice, which allows for significant growth and maturation of tissue structure. And then we went to see if neurons and glia would form a plexus. So just by section analysis, compared to the control organoids, which don't have any neurons or glia, we can nicely see down below where we've incorporated the ENS that neurons nicely form in between the layers of smooth muscle, shown in red. The neurons are in green. And we see not just enteric layers of neurons and glia, but we also start to see layers of submucosal neurons and glia. And these neurons can actually come together and form something similar to a small ganglia-like structures. But again, this isn't very satisfying in 2D. We wanted to see if they could form a plexus. So what we did, back to the movies, is we took a little biopsy from those in vivo grown organoids and did a whole mount immunostain. Now, to show that the neurons were actually coupling with the smooth muscle, what we did is we explanted one of those in vivo grown organoids and just put it in simple Ringer solution and subjected it to an electrical field stimulation with a lot of help from colleagues here at Children's to ask the simple question, when we stimulate the neurons, do those neurons then in turn go and trigger the smooth muscle to start to contract and relax? An organoid that again was grown on the kidney capsule that we've now triggered to see if the neurons in there will stimulate a peristaltic-like activity. Okay, so I'm now going to, so I just want to mention some of the things we're now using these organoids for. Of course, to study intestinal development. We've used them now to model Hirschsprung's disease, which is which in some cases is due to specific genetic mutations that affect neural crest development and differentiation into enteric nerves. But we're also using it to look at malabsorptive syndromes and developing transplant models, as well as to study enteric pathogens, which I'll touch briefly on in the next and second part of my talk, how to generate rather than generate intestinal organoids. Can we use a similar approach to generate organoids that are derived from the more anterior part of the GI tract? And this was work done and really pioneered largely by a grad student in the lab, Kyle McCracken, who became interested in generating a particular anterior GI derivative, the stomach. Now, the stomach is very different in humans than it is in mouse. This is a schematic of a human stomach from a review from Jason Mills and Ramesh Shivdasani. The human stomach is largely broken down into two domains. The corpus, that has fundic type of epithelium, which is involved in acid production and protease production, and then the distal part of the stomach, the antrum, around the pyloric region, which expresses many glandular epithelial cells, but has one cell type in particular that's unique to the antrum, the G cells, which produce gastrin, which is essential for proper digestion. Now, Kyle became interested in the stomach for a number of reasons. Firstly, the human stomach is vastly different than the mouse stomach. The mouse stomach actually has a whole segment called the forestomach that doesn't even exist in humans. It really, he wanted to develop a model to study specifically development of the human stomach. But another reason is that one of the most common bacteria to be in the stomach, Helicobacter pylori, does not model well in mouse. So Helicobacter is one of the primary causes of peptic ulcer disease and gastric cancer. And gastric cancer, while not as prevalent in the US or North America, is the leading cause of cancer-related deaths in other countries, particularly in Asia. So for the reasons that the mouse isn't necessarily the best model of stomach development and disease, Kyle felt it imperative to generate human gastric organoid models to better study human condition. So now he first set about by generating the distal part of the stomach, the antrum, and found a different set of manipulations that mimic early antral development were sufficient to push human pluripotent stem cells first in the endoderm, then into the foregut, which is the predecessor of the stomach and other organs, and then into the human antrum. And kind of a summary of the manipulations of that are shown here and in the next slide. Now, I'm going to briefly show that firstly, how Kyle was able to coax endoderm into the foregut rather than the posterior structures, the mid and hindgut. And that was predominantly through inhibition of BMP signaling, which at this stage of development, BMP is essential for making all the posterior derivatives, and inhibiting BMP with this molecule noggin was sufficient to push definitive endoderm into foregut. However, we still needed the Wnt and FGF to generate the three-dimensional structures. Kyle then generated posterior foregut through a brief manipulation of retinoic acid, and then continued retinoic acid treatment for another 3 days, followed by merely growth in EGF was sufficient for these early stage spheroids to grow out into gastric antral organoids. Now, what that looks like is shown here. These little structures are the early posterior foregut structures that actually grow out into, after a couple of weeks, into a simple epithelium, a cystic type of structure. However, over the next week or two, the epithelia starts to nicely form this glandular structure shown here, such that by the end of this in vitro culture, we see nice folds of epithelia that look, for all intents and purposes, like epithelial glands of a newly born mouse stomach. We know that these organoids are the antral part of the stomach because they express one of the key antral markers, PDX1, shown below here. So this is a section of a late-stage embryonic mouse stomach, and you can see PDX1 in red is not expressed in the fundic part, but is expressed in the antral part of the mouse stomach. Our organoids are definitively antral because they express this antral marker PDX1. Moreover, these organoids have all the different cell types that you would find in the early postnatal antrum of the mouse, shown here. On the left here is just a nice schematic of a stereotypic antral gland with the different cell types shown in different colors. The staining here is just showing the different cell types that we find in the pluripotent stem cell derived antral organoids. So the antral organoids have both surface and gland mucus cells. They have a nice progenitor zone shown by SOX9 expression here, right at the base. But importantly, they have one of the most definitive cell types of the antrum, and that is gastric-producing endocrine cells. And that shows definitively that these are antrum. Now, we also verified that these are not fundic by virtue of expression of the cell type that's in the most common to the fundus, the parietal cells. We know that there are no parietal cells in these antral glands by virtue of expression of the proton pump subunit ATP4B. So these are definitely antral organoids, but definitely not fundic. But that led us to the idea that, well, if these cultures are definitively not allowing for development of the fundic part of the stomach, how do we actually generate fundic organoids? There must be something that we're missing from these cultures that allows them to form the fundic epithelium. Now, this is where the paradigm of going back to the developing embryo kind of falls apart, because nobody really knows what the signaling pathways are that promote fundic development. So we were kind of back to the drawing board. However, through a nice collaboration with a number of students that were in the lab, particularly Julie and Baptiste, master's students from France, and Kaylin, we were able to identify that that Wnt signaling is absolutely essential for making the fundic part of the stomach. We went on to show through mouse knockouts and human foregut cultures that the fundic part of the stomach is essential for, that Wnt is essential for making the fundic part of the stomach, and that activation of Wnt in this window of time was sufficient to generate fundic organoids. So I'm going to quickly go through some of the mouse data, mouse and the in vitro data to show that. This is a dissected stomach domain from a mouse embryo, just showing that that gave us the first hint that Wnt signaling is important for fundic development. Using a Wnt reporter mouse, which basically will turn cells blue when there's high Wnt, we found that the fundic domain of the mouse stomach during embryonic development has high Wnt, whereas the antral domain does not. We then went on to test that the hypothesis that activation of Wnt can specify early fundic, going back to our human foregut cultures. And what we find is that when we activate Wnt signaling, we can turn our foregut cells into an early fundic stage organoid, shown by virtue of loss of the antral marker and activation of these fundic markers in green. And this is just confirmed here by immunostaining. We then went back into the mouse embryo and asked genetically, is Wnt signaling required for making the fundus? And in fact it is. So this is just one example of data from those studies. This is a section of a mouse stomach, and this is a wild-type mouse stomach, and the pink cells here are pink because they have beta-catenin protein in the epithelium, and these green cells are fundic cells, or are parietal cells in the fundic. When we delete beta-catenin, which is the key effector of Wnt signaling, throughout the stomach, what we see is that regions that have deleted beta-catenin, which you can see that the beta-catenin protein is no longer present in the epithelium. We can see that those regions can no longer form fundic-type epithelium. And if you just zoom in here, you can very clearly see that the regions that lack beta-catenin cannot make parietal cells. The parietal cells are marked in green here. However, regions of the stomach that failed to delete beta-catenin can very robustly make parietal cells. So pretty clearly, Wnt signaling via beta-catenin is essential for specifying fundus. So we then went on to following activation of Wnt signaling in our cultures to show that the organoids derived following Wnt activation are fundic in nature. So here are some examples. Again, these are fundic cell types and antral cell types. When we look at our now fundic type organoids, we can clearly see that they have all the fundic cell types you would expect, the mucus cells, the endocrine cells. Importantly, they do not have the antral specific endocrine cell gastrin. But more importantly is that these fundic type organoids have chief cells, which is one of the other cell types found in the fundic but not in the antral epithelium. So the chief cell markers, PGA5, PGC, and MIST1, these are pepsinogen genes, are very nicely expressed in the fundic organoids but not in the antral ones. By immunostain, we can see the fundic marker MIST1, the chief cell marker, and here we see patches of pepsinogen expressing cells. And here's a TEM done in collaboration with Taro Hara in Chip Montrose's lab at University of Cincinnati, nicely showing an example of a chief cell with packaged pepsinogen granules. Pepsinogen can actually, protein can be secreted from these as well. I will point out though that from a chief cell standpoint, these chief cells are still relatively immature and don't robustly make a mature pepsinogen protein. I should also point out that chief cells don't normally mature even in humans until well after birth. So this is consistent with these being more fetal in nature. However, these still did not contain parietal cells. Now again, the parietal cells are the cells of the corpus, the fundic epithelial cells that acidify in response to meals. So kind of back to the drawing board. However, with a lot of work from Kyle and the students in the lab, particularly Batiste Martin, we took sort of a low throughput screen for different signaling pathways that might be sufficient to trigger formation of parietal cells in our established fundic organoid cultures. And what we found is that a brief inhibition of MEK kinase along with BMP could very robustly produce parietal cells in these cultures, shown in this slide here. So this is a control culture without MEK kinase inhibition. Here, following 2 days of MEK kinase inhibition and BMP4 treatment, we see that the epithelial cells now robustly contain parietal cells stained in red. Moreover is that these organoids are patterned in more or less an appropriate manner when compared to the gastric epithelium. So here's a section from a human stomach with mucus cells at the surface and parietal cells down in the gland. We see that our engineered gastric organoids also have a similar architecture with parietal cells down in the gland. And when you compare either by immunostaining or by TEM, you can very clearly show that these are very similar to bona fide parietal cells. However, the real test is, can these parietal cells secrete acid? Which they can, shown in this slide here, again done with great collaboration with our colleagues across the street at the University of Cincinnati, Taro and Chip. So the top circle, so on the y-axis here is measurement of pH in these gastric organoids by a pH-sensitive dye. And you can already see that antral organoids, the open circle here, have a pH of about 7.2. Now parietal cells already have a more acidic pH, just at baseline. However, one key hallmark of a parietal cell is that they acidify in response to histamine. Now, histamine is released after you eat and is one of the key triggers of acidification in the stomach. So what we did is we treated these fundic organoids with histamine, and within about an hour or two, we saw a pretty decent drop from pH 7 down to about 6.6 in response to histamine. Now we know that this is acting through histamine receptor because we can block histamine receptor with this antagonist, which is one of the common drugs that people take to reduce acid production, famotidine. And we can see that famotidine blocks histamine-induced acidification. We then validated that this was acidification of compartments using a separate approach, just to show that in fact in response to histamine we get very nice acidification of secretory compartments in these organoids. So I think the gastric organoids are going to be a really great tool to study how different cell types in the stomach emerge developmentally, but also we're in the process of using them to study now more applied aspects of stomach biology. And one example that I'll show is we're using organoids to study pathways that are triggered by H. pylori. Now, and this is done as a great collaboration with Jana Zavros and the folks in her lab. Now, H. pylori is a bacteria that's found in most of us, yet in some cases H. pylori colonization over the long haul will trigger activation of a number of downstream pathways in a gastric cell. And how it does this is it binds to the surface of an epithelial cell and injects pathogenic proteins into the cell. One of these proteins is called CAG-A, and what CAG-A does is it physically interacts with several different proteins that regulate signaling in the gastric epithelial cell. One example is shown here, the c-Met tyrosine kinase receptor. It also acts on a number of downstream pathways, but at the end of the day, what CAG-A does is it can trigger aberrant proliferation of the gastric mucosa, which is, of course, is an early stage indicator of gastric disease. So we wanted to see if this process actually happened in our in vitro derived organoids. So what we did is we made organoids, brought them over to Jana's lab, where her graduate students, Mike, in this case Mike Shoemaker, would inject H. pylori into the organoid. Now what we saw was H. pylori very nicely bound to the inside of the epithelium. Here's just a high-resolution electron micrograph showing that, and it triggered the expected downstream effects. Now here you see that only pathogenic H. pylori, G27 here, would trigger phosphorylation of the c-Met receptor, and would actually do this probably through direct interaction with the c-Met receptor, shown in this immunoprecipitation pull-down. However, non-pathogenic forms of H. pylori wouldn't do this. Also, the pathogenic H. pylori would very quickly, within a day, trigger a doubling in the proliferation rate of the epithelium. However, non-pathogenic H. pylori would not do this. So what this says is that we now have a pretty minimalistic system to really look at the effects of H. pylori directly on the gastric epithelia. So we can model the early stages of intestinal disease in this in vitro system. Now importantly, for the folks in the audience who appreciate that most of these diseases come along with a chronic inflammation, what we're in the process of doing is now adding inflammatory cells into these organoid systems to then mimic the other key driver of early stages of disease, and that is chronic inflammation. So we can really dissect the different contributing factors, the bacteria versus the inflammation, on how they drive derangement of the normal epithelial homeostasis. So I'm going to finish up now. So I went a couple of minutes longer than I had planned. Just to say that these organoids are great to study development and how cells emerge during development, but they're also, I think, an emerging model to study disease processes. I did not talk about that they're actually great models to study mechanisms of epithelial regeneration, as I mentioned, infectious diseases, and the Cleaver's lab have nicely shown organoids are great cancer models. But I also want to highlight that these PSC-derived organoids are really, in our hands, I think, emerging as a great model to try to engineer functional tissue that we think we, in the future, might use in transplantation-based approaches. I think I've thanked everyone along the way. Hopefully I have. And I want to end by just saying that a lot of what we can do is because we have a really robust pluripotent stem cell facility here, which provides embryonic stem cells or generates iPS lines, can do CRISPR editing, and provides training courses on stem cell manipulations. And I'll stop now and take questions. Thanks.
Thank you very much, Dr. Wells. That was a very engaging talk, and really impressive to see your work on how you created both the functional ENS with intestinal tissue and also developed these human gastric organoids with the disease modeling as well. We'll now move to our Q&A session by addressing some of the questions that were submitted during the presentation. Let me just start out by asking a little bit more about the intestinal organoids, because there was quite a bit of interest in these. So from from the grafts you showed into mice, can you also transplant these organoids into human?
Well, in theory, you could. You know, that's going to take more development of larger segments of bowel, and of course, showing safety and efficacy in animal models, which we're in the process of developing these transplantation models directly into the intestine to show that they can function. And again, this is done through collaboration with Mike Helmrath, who is a pediatric surgeon. In the future, yes, of course, we want to generate segments that can be transplanted into humans, but that's quite a ways down the road.
And I guess with that, because the organoids you have developed are somewhat immature or of a fetal state, what is the best method to mature them, do you think? Is that in vitro or is that in vivo, like transplanted into the future patient, but for now at least the mouse?
Both will work, but certainly the transplanting them into an animal seems to provide the best maturation. In part, basically because they're getting, you know, a constant blood supply, so all the nutrients and oxygen that they need. The limitations in vitro are that they can only grow to a certain size before they start to become necrotic, because you can't get oxygen to the inside of these organoids effectively. We are very interested in developing fluidic platforms that would allow us to get better oxygenation and nutrient delivery to in vitro derived organoids, with the goal of improving their growth, but that's also work in progress.
Thanks. So just looking here, I have a few questions on more specifically on the organoids here. So one is how long they can be cultured in vitro, and another one is what are the differences in the organoids that's like when you create like a very small piece of organoid, and then when you have and you transplant that versus if you have a more developed one with the ENS and a more developed blood supply, like how do they function differently?
So the small intestinal organoids that I showed at the beginning can be cultured for, we've done it up to a year. That was in the original STEMCELL et al. Paper. If they're grown in a high wind environment, they can be passaged mechanically for up to a year. In terms of ones that the differences between the organoids with and without an enteric nervous system and how they grow, they're actually quite different. Insofar as the enteric nervous system, incorporating the enteric nervous system into these organoids helps other cell types grow better. For example, we get better maturation of interstitial cells of Cajal when we transplant, when our organoids contain an enteric nervous system. We also get probably better vascularization when our organoids contain an enteric nervous system. So, and that's not surprising given the important role that the ENS cells play during development of the intestinal tract.
Thanks. Just, I have a question here. It's very, very broad, but do you have advice for researchers developing organoids for other tissues who currently don't have an organoid model? What would be the next best after organoid models? I assume the question is kind of leading to.
Right. Well, I mean, for us, again, you know, you have to decide what you want to use them for. There are now gastrointestinal organoid models of most regions of the GI tract derived from adult patient biopsies. So there are organoids of the esophagus, of the stomach, of the small and large intestine. Again, if you have access to human surgical samples, you can use one of the published protocols to try to use organoid models from them. In our case, as I described in the talk, we're generating new organoid models through directed differentiation of pluripotent stem cells. And for us, that really depends on understanding the normal stepwise process by which organs normally form. And we've run into problems trying to generate other organoid systems such as colonic or esophageal because there isn't as much known about the normal development of those organs. So it's taken us much longer to develop other organoid systems for organs that we just don't really know much about how they normally form. So, and as I said, the organoids that we develop, since they have more complexity, might be better for certain applications. However, the adult-derived organoids that only have an epithelium might be easier to come by and might be better for modeling other aspects of epithelial biology. So you really have to define what your question is before you kind of invest in one organoid approach versus another.
And I guess with that, organoids are maybe not also everything. I mean, 2D model systems are still very valid to answer some of those questions. Would you agree with that?
Yeah, no, I would definitely agree. The main disadvantage to most 2D models is that they often start with transformed cell lines. And of course, we all know physiologically those aren't the best. I should mention that there's quite a bit of effort being put in to taking three-dimensional primary organoid tissues and transferring them to 2D models because there's a lot of great things you can do in 2D. Looking at epithelial barrier function and secretions and things like that work much better in 2D. So, but I still would say that it's best to start with primary cultures of actual GI tissue rather than using a cancer cell line. I would tend to think that the primary tissues will ultimately be more informative. But I agree, there's a lot of uses for 2D cultures, and there are new emerging models of 2D systems coming down the pipeline from a variety of labs.
Yeah, thank you. If we move a little bit into the gastric organoids you showed, I think a lot of people would like to know a little bit more about the H. pylori injection model and really understand how that works. So one question is, how long can you keep the H. pylori inside the gastric model and work with it?
Yeah, no, that's a good question. When we inject bacteria into these GI organoid models, and you know, people have done this now with other pathogenic bacteria or even commensal bacteria, usually limited to a couple of days because the bacteria, while they are contained in the lumen of the organoid, you don't actually get overgrowth of the outside of the organoid typically because you can include antibiotics that can't get into the lumen. So the bacteria in the lumen can grow, but no bacteria grow outside of the organoid. So that's worked now in a lot of people's hands. However, once they get into the lumen and if they like it and they start growing, they basically will use up all the oxygen and these are pretty terminal experiments. So I would say even when you start with low numbers of bacteria, you're still just looking at, you know, a 1 to 4 day type window to do these experiments.
Yeah, so you are somewhat limited there. And how do you think you can apply this system to drug discovery, more specific?
Well, of course, you know, the last example I showed where, you know, the proton pump responds to already commercially available drugs, it seems that these organoids can respond in a stereotypic fashion to existing drugs. So people have used organoids for cystic fibrosis-related drugs and other things. So I think as we move on and have new needs for different types of drugs, the physiologic response in these organoids is going to be, I think, pretty good. So of course, for us, using organoids that contain an enteric nervous system might be a good screening tool because the enteric nervous system actually is often hit through off-target effects by new drugs. So if we could look for off-target effects on the ENS using this organoid system, it would save a lot of time before going into clinical trials. So just even looking for something as simple as off-target effects, I think these will be pretty useful.
Thank you. We have a lot of questions, but we are running out of time. So I think I'll just end with this one question that I think also a lot of people are wondering about. What were some of the biggest challenges your lab faced when developing gastrointestinal organoids in vitro?
Well, probably the kind of the real shift came when we figured out how to transition two-dimensional cultures to three-dimension. You know, you can't have an organoid without three-dimension, and I think that discovery was, as a hurdle, really allowed us to now generate three-dimensional organoid systems, you know, all along the GI tract. So I think really learning how to transition these tissues into and to coax them into forming 3D structures spontaneously was really kind of an aha moment. From that point, it was more or less trying to figure out how to drive them into specific regions of the GI tract, and that's really an ongoing challenge. But I think we're coming along pretty well.
Thank you. I think you are doing pretty well too. So unfortunately, that's all the time we have today. I'd really like to thank everyone for attending, and especially thank you, of course, Dr. Wells, for your time with us today. I'm looking forward to continue to follow your work and see where you take us next. Thanks again, and goodbye, everyone.